Buenas tardes a todas las personas que nos acompañan en esta sesión. Una maravillosa ocasión que tenemos la tarde de hoy para escuchar a la doctora Emma Kowal, que nos acompaña desde la Universidad de Deakin en Australia. Y vamos a tener una conferencia con ella titulada El fantasma de la ciencia racial en la genómica indígena. Esta conversación con la doctora Emma ha sido fomentada por el doctor Francisco Vergara del Instituto de Biología de la UNAM y también por la doctora Ivette García Deister de la Facultad de Ciencias de la UNAM. Y aquí en la sesión de Zoom nos acompañan algunos amigos cercanos de los proyectos y también asiduos participantes al ciclo de conversaciones sobre raza, racismo y antropología biológica, que no ha tenido sesiones este año, ya sé que las extrañan, pero ya tendrán noticia de ello. Y este espacio, pues de alguna forma es una continuación de varias de los temas e inquietudes y líneas que ya hemos trabajado desde hace varios años en Surge y particularmente con Francisco con Vivet. Entonces, voy a hacer una pequeñísima introducción en inglés para darle la bienvenida a Emma y le voy a dar la palabra a Francisco para que nos las pueda presentar formalmente. Saben ustedes que quien nos acompaña en YouTube, como siempre, podemos leer sus comentarios, sus preguntas, todo estamos como muy abiertos a conversar. Entonces, una vez más, muchas gracias por su asistencia. I'm very glad to welcome Dr. Emma Cowell from Deakin University in Australia. We've been like exchanging emails for a while, and finally we have this space to hear her talk, The Ghostly Presence of Race, Science in Indigenous Genomics. We're going to have this conference in English, initially hoping that we can translate it in the future for the YouTube channel. But well, any question or comment in Spanish or English is welcome here in the Zoom session or in the YouTube transmission. Thank you very much to the SAIC for the support of the transmission. And well, I give the word to Francisco to hear the formal presentation.
Muchas gracias, Jimena. Thank you, Jimena. Welcome, Emma. Welcome, everyone, to this session. We are really looking forward to listen to a presentation that will mostly deal with the recent book that Emma has published. It's really a wonderful work. It comes from research that has taken her decades. And so to start, I am going to read a formal presentation, like a biographical sketch. And then after that, we will hear Emma talk for approximately 45, 50 minutes, maybe. It is possible, we have just asked her that anyone who has any question or wants to stop Emma for any reason related to the content of what Emma will be presenting. This is allowed, this is possible. And after Emma is done, Ivette Garcia Deister will offer a comment on the book. And she will also talk a bit about a research group that she is currently coordinating. And then we will start the session of questions and comments from everyone, including people who might be joining us on the YouTube transmission that is already on. So Emma Kowal is Distinguished Professor of Anthropology and co-convener of the Science and Society Network at Deakin University, Australia. She's a cultural and medical anthropologist who previously worked as a medical doctor and public health researcher in Indigenous health. Her research interests lie at the intersection of anthropology, science and technology studies, or STS, and Indigenous studies. She is an award-winning researcher and educator, past president of the Society for Social Studies of Science, the 4S, and a fellow of the Academy of the Social Sciences in Australia and the Australian Academy of Health and Medical Sciences. She has authored 150 publications, including the monograph Trapped in the Gap, Doing Good in Indigenous Australia, and the collection Cryopolitics, Frozen Life in a Melting World, and her latest book is Hunting Biology, Science and Indigeneity in Australia, published by Duke University Press in 2023. Emma, the floor is yours. Thank you.
Thank you so much. Thank you, Jimena. Thank you, Paco. Thank you for this invitation to be here. I wish I could be there in person, but this will do. And it's really an honour to be talking to the Surge audience. And I'm so looking forward to to being in conversation with you. And particularly as what I have to speak about, I know has huge resonances for scholars in Mexico and in Latin America more broadly. And I'm really grateful that I can just speak in English here. I don't speak Spanish, so that's their only option, but I just very aware of that linguistic privilege that I can go around the world speaking English. And I do, and I hope that my accent is not too disorientating for people. So I'd like to start. So this is start by acknowledging the traditional owners, which is something that we do in not acknowledgement of country. I think it's quite common, I think now in North America and Canada, but it actually started as a kind of post-colonial phenomena more in Australia in really way back in the 1980s. So I acknowledge the traditional owners of the lands where I am at the moment in Melbourne in Australia, the Wurundjeri people of the Kulin Nations, and acknowledge their elders and their custodianship of the land. And also I have here a warning which is appropriate for an Australian audience, but I've just kept it here, that there are images of Aboriginal and Torres Strait Islander people who have passed away. And there's cultural taboos in some groups about seeing images of people that have passed away, that have died. And also there's historical terms of using at times with outdated terms for Indigenous people that would be inappropriate, and to be using those terms apart from this academic context. Okay, so as Paco mentioned, I'm drawing today on some material that's in the books Haunting Biology, Science and Indigeneity in Australia that came out a year and a half ago now. And I'm going to first give some background on the history that the book covers. Some of it will be extremely familiar to some of you, others it may be new, and I'm giving the history, of course, with an Australian lens. And then I will talk about one of the stories in the book in more detail, which does involve this particular hair sample. So remember that, because we'll talk about that again. So Indigenous biological knowledge was one of the very first exports from the newly colonised Australian continent, very shortly after the initial British settlement or invasion, as it's called by others, in 1788. So we're very kind of late date of colonisation. Doctors, amateur anthropologists and government officials sent Indigenous remains to leading comparative anatomists such as Johann Friedrich Blumenbach. So from the very, very first years, Indigenous remains were recognised as an important commodity that needed to be spread into the metropole. In America, Samuel George Morton systematized skull measurements by filling them with shot pellets to measure their internal capacity. This is this IC in this kind of iconic table from one of his very important popular books. So he produced tables of this biological data that mirrored the racial hierarchies of the time. Australians often appeared at the bottom of the list, as we can see here, above only the Tasmanians, who were considered a separate race. So Tasmania is an island at the bottom, at the south of Australia, that is part of Australia, but racially, people there were considered to be a different race to the rest of the Australians, and the most primitive, or as people would speak about that at the time. So later in the 20th century, later in the 19th century, the spread of evolutionary theory saw Indigenous Australians as living remnants of Stone Age man, the missing link between humans and non-humans. And then in the 20th century, as the 20th century dawned, a new kind of collecting emerged, which was fieldwork. And that term fieldwork actually was first coined by this particular expedition. You can see the picture of the 1898 Cambridge Anthropological Expedition to Torres Straits, led by anthropologist Alfred Court Haddon, the guy that's crouching down. He was a British ethnologist, and this was a really important expedition in the history of anthropology and really in all of kind of human sciences. So what was really important in this expedition was that rather than relying on the reports of colonial correspondence, of people writing back to the ethnologists in their armchairs in Europe, these ethnologists and physicians began collecting their own data from living subjects, anthropometric measurements of faces, limbs, hair, and skin. Another really important episode in this, which comes up in the book a lot, is called the Board for Anthropological Research, which was based in Adelaide, which is the south of the middle part of Australia. And they began expeditions in 1927 that continued until the 1960s. They had multidisciplinary research teams, including dentistry, physiology, and psychology. And here's just a few images from those expeditions, which again feature quite a lot in the book. In the early decades of the 20th century, so shifting now a little bit further again, there was great hope that blood group research, the new field of serology, might provide an objective measure of race. Research conducted during World War I in the port of Salonika, because it was a World War I port with soldiers from all around the world, and there were some researchers there who were opportunistic and thought, well, let's compare the blood groups of all of these people, from these soldiers from different places around the world. So that kind of was the beginning of using blood groups to look at human difference. And they found that blood group A was predominantly found in Europeans, and blood group B was found in Asians. So then, similarly to what we've seen with DNA research in the late 20th and 21st century, back in the early 20th century, blood group research then became the international trend, and everyone was looking at, well, what are the blood groups of the particular Indigenous people where we live? So in Australia from the 1920s, Adelaide-based scientists led blood group research, and interestingly, they were expecting that there would be B group blood, but they did not find any blood group B group blood in Indigenous Australians. And so this was used at the time to support this theory that Indigenous people were actually what they called archaic Caucasians. So even though this sounds extremely, parochial maybe, but it was a very much a international, internationally dominant theory of the origins of Indigenous people, that they came from this group of the proto-Caucasoids that both gave rise to Aryan Europeans, all of these other particular groups, including the Ainu, Indigenous people in Japan, the Vedas and Dravidians in India, and then also Indigenous people. So Indigenous people were seen at that time, and this is like late 19th century into the mid 20th century, as archaic Caucasians or primitive cousins of present-day Europeans. And this was used to justify or to explain the relative ease of colonisation and miscegenation in Australia in quite interesting and at times disturbing ways, which I discuss in a chapter of the book. So then, moving forward again a bit through time, by the middle of the 20th century, technological advances in serology and particularly in technologies of freezing, which is another issue I've always been very interested in. There's a book I've edited with Joanna Radin called Cryopolitics, which looks at freezing, but freezing comes into this story where people were able to take blood samples, freeze them, and send them back to the metropole, which in this case, this man on the left, a guy called Bob Kirk, who was a British geneticist who moved to Australia in the late 50s and started to collect blood samples from Indigenous people here in the north of Western Australia. And then he would take those samples back to his world-class starch gel electrophoresis laboratory for those history of science, 20th century science nerds. So this is using potato starch from Canada to separate out the proteins in these blood samples and looking at protein variants within them. So biologists believed that the long isolation of Indigenous people on the Australian continent would mean that their bodies contained many unique proteins. And this still very much persists. And even though theories like the archaic Caucasian are completely, discredited, no one talks about that anymore, but the idea that Indigenous people have been on the Australian continent for a very long time. In the 80s, people used to say for 40,000 years. That was the slogan that Indigenous people would use. We've been here for 40,000 years. It turns out that was the limit of radiocarbon dating. So the number came from a technological limit. So now with ancient DNA, which is another issue that I'm quite interested in, we know that the current number is around 65,000 years that people are talking about, that Indigenous people have been on the Australian continent. So whatever the actual number, the idea that Indigenous people have biological unique characters from their isolation, that has been very consistent across time. And most famously, and there have been a lot of interesting discoveries from science from Indigenous bodies, and probably most famously was Barry Bloomberg, who Bob Kirk, the scientist I just mentioned, supplied him with serum from Aboriginal people in Central Australia in the desert, and that's where Bloomberg found what he initially called the Australia antigen and was later identified as hepatitis B. And he got a Nobel Prize for that. So hepatitis B was discovered from one of these blood samples. So that's been like a very brief tour around some of these important points in the history of making biological knowledge about Indigenous people in Australia. So drawing on both some famous episodes and some very obscure episodes in the history of scientific research on Indigenous bodies and Indigenous populations, my book explores wider questions about the production of biological knowledge. I ask, how are we to understand Indigenous biological difference in the 21st century? Is it a racist ruse, a stubborn residue of racial pseudoscience? So is biological difference just something that is still hanging around from those days of Samuel Morton's tables of internal capacity? Is it something that exists but that should not be allowed to have social or political relevance? So maybe Indigenous difference or just biological difference does exist, but we should not get so caught up in it, but and just make sure that it doesn't lead to discrimination. Or is it a potentially empowering force that can be unlocked by newly accurate science and by being under Indigenous control? So these questions that I'm really grappling with, and to answer these questions, the book traces the rise and fall of different kinds of biological and medical inquiry over the 20th century and up to the present. So some of that history that I've just told now. Each new discipline claimed that new theories or methods that were superior to the previous modes of knowledge production were going to actually finally find the answer, whether that was craniometry or serology or now genetics. And along the way, thousands of bones, hair samples, blood samples, pathology slides, samples of placenta, and much more were acquired, collated and stored in museums and laboratories across Australia and the global north. The material persistence of samples over decades and centuries, the fact that a lot of these samples still exist, this folds together the fates of different scientific methodologies and racial theories across time and space. And I'm really influenced there by Ahmad Macharek from Amsterdam and her work on time and folding and DNA. So my path to writing this book, because that's important, like why did I write this book? What was I trying to achieve? How did I come to be asking these questions that I'm asking? It's wasn't an accident. It's a story that has come through my long involvement in 21st century genomics in Australia. For a long time, genetics and genomics were taboo subjects in Indigenous circles because of their historical association with damaging racial hierarchies. So I began working in Indigenous health as a young doctor over 20 years ago. Everyone I knew then considered the idea that Indigenous people might be genetically susceptible to some diseases to be racist. So I don't know whether that kind of taboo also was in Latin America at that time, but definitely it was in Australia. To say that, maybe Indigenous people have different alcohol genes that mean that they're susceptible to alcoholism, that would be absolutely only something that someone who was either who was racist or who was not aware that that was making them seem racist would say. And this was soon after the Human Genome Diversity Project, which I know many people here are aware of. So this was a companion to the Human Genome Project in the 1990s. So the Human Genome Project sequenced the genome and has led to a whole lot of investment in and scientific discovery. The Human Genome Diversity Project was alongside that much smaller project led by geneticists, population geneticists, and it was soon met with organised resistance from Indigenous groups, particularly in Latin America, of course, but also in Australia, who dubbed it the Vampire Project. And the Indigenous Peoples Council on Biocolonialism was formed partly in response to that, but also the bioprospecting through organisations like RAFI, which have been incredibly foundational in this movement. So in Australia, no Indigenous people participated in the Human Genome Diversity Project, and it was seen as, you can never do genetic research in Indigenous communities. So that was going on then. At the same time, I was working on my first book that came out of my dissertation, and that was focused on the ways that non-Indigenous people construct the idea of Indigenous cultural difference. And I did my fieldwork for that project in the mid-2000s, working with Indigenous health researchers in the north of Australia, where I also was living and working in Indigenous health. So it was a native ethnography in that sense, that I was studying my own tribe of non-Indigenous workers, researchers in Indigenous health. And when I completed the work for that book, the question of biological difference began to interest me. And that was because even though there was a really strong taboo against genetic research that I mentioned, huge investments in genomics through the Human Genome Project, but continuing on, meant that it would eventually come to bear. It would eventually have an impact on Indigenous research. I also became really interested in science and technology studies, which has been become, along with anthropology, my kind of disciplinary home. That had many scholars working on historical and contemporary questions of race, science, and colonialism. It seemed to me that the rise of genomics would inevitably collide with the stigma of biological difference in Indigenous research. So I thought, well, if we analyse this collision as it's happening, as genetics kind of reaches Indigenous communities, that would tell us really useful and important things about 21st century intersections of race, science, and justice, which are my abiding interests. So, over the last 15 years, the taboo on studies of Indigenous biological difference has definitely been lifted, and it's been quite incredible, actually. Genomics has become a common tool in studies of Indigenous health, in ancient DNA, and in direct-to-consumer genomic ancestry testing, which I suppose are three kind of broad areas where I've been following these questions of indigeneity and genetics and biology and difference for many years now. And rather than a passive observer, I've been an active participant in these shifts. So I definitely have, skin in the game, we'd say in English. My background in Indigenous healthcare and research, combined with a history as a Indigenous solidarity activist, led me to support national conversations among Indigenous leaders about how genetic research should be governed. So, long story short, I became also had a big part in setting up the National Centre for Indigenous Genomics at the Australian National University, which is the first Indigenous governed genome facility in the world and is still very much going strong. And I also have founded the Australian chapter of the Summer Internship for Indigenous Peoples in Genomics, Sting Australia. And I know that Jocelyn, a colleague working with you, has done incredible work to set up Sing Mexico. So that's really exciting. So this experience as both an anthropologist of science and indigeneity, and as a participant in Indigenous governed genomics, has taught me about the importance of careful histories of 20th century biology. There are resonances and connections between older sciences of human difference and work today on precision medicine, pharmacogenomics, and a new field called evolutionary medicine, which is like both new and old and really quite interesting, but we'll save that for another time. So to understand these new areas, no one who makes knowledge about Indigenous people in the present is immune to these legacies of the past. But it's equally important not to essentialise contemporary science as a simplistic continuation of 19th century racial science and all the negative social consequences of eugenics and indeed the Holocaust, which is, always like the unspoken referent for eugenics and unethical research. We need analytical dexterity to trace the changing scientific conceptions of race across the 20th century, rather than conflating or collapsing different disciplines, tools, and theories together. So these things are not just the same at all. My aim is not to accuse 20th or 21st century biology of being just like 19th century racial science, but rather to understand the rise and fall of different disciplines within a longer story of making biological knowledge about Indigenous Australians. And just the longer story of human difference and how we understand and encounter difference. So I want to shift now to talk about a specific story. So are we going okay? Yep. All right. So I'll just tell this specific story to kind of try and give you a more concrete example of how these kind of dynamics play out in the stories I tell in the book. And then this is the second half of the talk. So this story is about a hair sample, as I mentioned. So I suppose hair. Does that make? I'm always aware that people are used to hearing American accents. So hair, but I say hair in my Australian accent.
Cabello.
Good. So this hair sample began its life on the head of a person, as most hair lives. And it was on the hair of a person in the Goldfields region of Western Australia in September 1923, when an ageing British ethnologist encountered a young Aboriginal man. They met at a place called Golden Ridge Station, 20 kilometres east of Kalgoorlie, which is in remote Western Australia, and named after a short-lived gold mine. So this is probably what it looked like, a railway siding on the Trans-Australia Railway, which had just been built in 1917. So Kalgoorlie is very remote Western Australia. There are a lot of gold mines. Basically, it still is the site of a lot of gold mines, and Golden Ridge Station is just like the second last stop along. That's where it is on Google Maps, Golden Ridge Station, and that's the satellite picture. So it's now an abandoned gold mine. And it had only been completed, the railway, 6 years earlier before this happened. And the two people that met were someone I mentioned earlier, Alfred Court Haddon, who was the Cambridge zoologist turned anthropologist who led the 1898 Torres Strait expedition. He was a leading scholar of race in the early 20th century, and he believed that hair was the primary marker of race, and he collected thousands of hair samples in his lifetime. So he was really interested in hair as well as bones and everything else. You can see this kind of obligatory skull in people's portraits at that time. The railway that came through brought Indigenous people together with access to travellers, to Westerners, to white people for many areas who had very little contact with white people prior to the railway. And by the time, this is just an image from during the construction of the railway, and along the railway stops, these became sites of intense trade and other forms of encounter between Indigenous people, passengers, and railway workers during their brief stops. So a lot of things were exchanged. but hair would have been an unusual trade item. It's likely that Haddon offered money, food, or tobacco to the man in exchange for snipping. This is the card that he wrote to go with the sample. One complete double lock and one short lock, and adding it to his luggage. So he snipped off the hair and during this short visit at the railway siding. Haddon at that time was 68, so he was near retirement and in Australia for the second Pan-Pacific Science Conference, which was held in Melbourne and Sydney, the colonial capitals. The conference featured the discourse of salvage anthropology. Delegates were urged to contribute to the task of salvaging Indigenous knowledge before it was too late. It was a very common phrase. And Haddon, he didn't collect any other hair samples on his journey that I can tell. He was on his way home. And I imagine what happened here was an opportunistic collection, the convergence of a persistent collecting impulse and a particularly impressive head of hair. So for Haddon, hair was a crucial tool for classifying human races within the natural sciences of the 18th and 19th century. And I think this is one area just within the history of science that has been a bit neglected. We normally think about skin colour, but hair was just as important, or even for some scientists more important. It had many advantages. It was easy to observe someone's hair from a distance, easy to collect and transport, and well preserved over time. So these categorizations of hair, in which you can see here in the contents of Haddon's important book, The Races of Man, they derive from the French naturalist Bory de Saint-Vincent, who first proposed a dual classification of léotrique for straight-haired or holotrique, woolly-haired. That was in the early 19th century. Another guy, Pruner Bay, the German 19th century physician, uttered perhaps the most often cited statement about hair, which is quoted in Haddon's book. A single hair presenting the average form characteristic of the age might serve to define it. And this kind of comes up again and again in this literature at the time, and is very much a kind of encapsulates the idea of race. So Haddon collects his hair, goes back to Cambridge, and his objects, not just the hair, but thousands and thousands of objects that he collected, both cultural and physical, biological objects, made a major contribution to setting up the Cambridge Museum of Archaeology and Anthropology. In 1945, most of the hair and skeletal material from the museum, including Haddon's collection, was moved across Cambridge, across the campus, to the newly established Duckworth collection, which focused on biological anthropology. And so the hair sample stays there in the now called the Duckworth collection, and it stays there for about 88 years. So meanwhile, time passes, samples persist, and things change. And now we get to the late 20th century, where advances in DNA extraction and amplification techniques opened up new fields of inquiry into human variation and evolution. Scientists began to extract DNA from recent and mummified museum specimens. And this was the very first one in 1984, when they managed to get DNA sequences from an extinct horse. After the development of the polymerase chain reaction, PCR, in the late 1980s, ancient DNA scientists began to get more and more DNA out of more and more materials. By the turn of the 21st century, aDNA had become a prestigious area of science, populated by evolutionary molecular biologists, anthropological geneticists, and paleogeneticists. In 2005, the the development of massively parallel sequencing or next generation sequencing emerged and was immediately taken up by ancient DNA researchers. So as these DNA methods rapidly developed, so did the political and ethical environment of the late 20th century. And I'm talking here about the Indigenous repatriation movement. So at the same time that the Human Genome Diversity Project that I talked about earlier made blood samples more problematic to maintain, the efforts by Indigenous people to get bone samples or just any biological samples related to their ancestors returned meant that those samples were also more difficult to get. So this is a famous Indigenous activist at the British Museum getting some samples returned through the repatriation movement. So the combination of these political and ethical shifts was to make first bones and also blood originating from Indigenous people less available to scientists. So it makes sense that some ancient DNA researchers would look to other less controversial biological substances to access. And maybe it was inevitable that hair samples left over from anthropometric studies would eventually attract the attention of DNA researchers. The geneticist who won the race, because these geneticists are extremely competitive, so the guy that won the race to extract more hardy mitochondrial DNA from hair, so that's mtDNA, was Alan Cooper, a New Zealander and former student of leading evolutionary geneticists Svante Pääbo and Alan Wilson. In 2004, Alan Cooper's lab at Oxford extracted mitochondrial DNA from horse hairs, 64,000-year-old bison hairs, the hair of an Andaman Islander. The Andaman Islands have also been a very kind of iconic place of like so-called primitive people, and 6 samples that were supposed to be from Isaac Newton, but at least 3 of them were not. So once this guy had sequenced mitochondrial DNA, the more hardy, smaller bits of DNA from hair, the race was on to sequence the first ancient human genome. That is information from all of the DNA, the nuclear DNA, from a hair sample. And it was won by this guy called S.K. Willeslav, who was a former postdoc in Alan Cooper's lab. And he is extremely famous in Denmark and within the field of ancient DNA. And he, in 2010, he was the first person to sequence a genome from a hair sample, which was from a 4,000-year-old man from the Sakak culture of Greenland. So this encounter I described at the start of this part of the talk at Golden Ridge Station in 1923 would have remained an obscure moment within a very long collecting career, if not for Willeslav, this guy, going to the Duckworth collection in 2010 to find a hair sample, just to find hair samples that he could try and get DNA from. And he chose the Golden Ridge sample as it was the largest one with the best chance of extracting DNA. He was advised by the head of the collection, of the Duckworth collection, that the Indigenous tribe from where the hair originated had died out. So he was told that there was no one he could consult with. And the ethics committee at the University of Copenhagen, where he was, did not require ethics approval because it was not research on living people. So he just took the sample to Copenhagen and extracted the DNA. So his team extracted DNA to the depth of 6.4 times, which is quite impressive, particularly for that time. And they analysed it and they concluded that the population that would become Indigenous Australians split off from the population that would become Europeans and Asians around 70,000 years ago, which fit with the archaeological record at the time. So this lock of hair was elevated to iconic proportions within the scientific community in 2011 as the source of the first Aboriginal genome ever to be sequenced. The scientific achievement was matched also with an unexpected advance in ethical practice. So this is one small detail about the process of publication that the paper was initially submitted to the journal Nature, but when it came out, it was in the journal Science, which seems like a very small detail, but actually reveals a whole story beneath that. So basically what happened, and I'll kind of try and do this briefly because despite saying I go for no more than 40 minutes, I think I'm pretty quickly getting to 40 minutes.
It's fine. You can go ahead, please.
I'm fine. All right. So this is like this story of how this paper ended up in Science instead when it was submitted to Nature. That small detail actually tells us a whole lot about what I call, or about what Karis Thompson explores as good science in her book, which I have a slide of in a little bit. So basically, the main part of what happened as why it was submitted to Nature but came out in Science was the reviewers. The research was completed with no Indigenous involvement whatsoever. As I said, he was told, S.K. Willisler was told by the collecting institution that there was no one to consult with because the Indigenous people had died out, and he was told by his ethics committee that he didn't need ethics approval because it's not on living people. But the journal reviewers and then subsequently the journal editors raised a crucial question. How is it that this groundbreaking research with huge potential implications for Indigenous people could occur without talking to any Indigenous people, particularly when using a specimen that was collected in circumstances very different to contemporary standards of consent, and also that many groups would consider to be a human remain, that a hair sample, so whether a hair sample is a human remain that needs to be repatriated is an open question. Sometimes it's considered to be human remain and sometimes it's not. So this small detail of which journal it came out in, we can think of that using Karis Thompson's notion of good science from her study of stem cell science. So what is good science? A good scientific experiment is well designed and answers the questions it sets out to answer, or leads to further, even more productive questions. So it's good, it's well designed. We could think about good science that it's good and it's not bad in terms of it follows the rules. So it conforms with ethical requirements, it meets disciplinary and institutional standards of research integrity, such as accountability, transparency, replicability, and so on. But the way that Karis Thompson wants to explore the idea of good science, which I think is really useful here, is it's a descriptive term. It's not a normative term, but a descriptive term for fields that raise complex societal issues and so develop their own ethical apparatus to engage interest groups beyond science itself. So stem cell science, that she studies, it's a science that has ethics. It raises particular ethical issues. So I think that's a really interesting way to think about research on Indigenous Australians and genetic research broadly. Research with Indigenous Australians has ethics, at least since 1991, when the first specific ethical guidelines for Indigenous research were produced. So this is, quite early. A lot of places barely had introduced informed consent forms at all. And in Australia, we already had specific guidelines. These require researchers to demonstrate that their work actively involves Indigenous communities and is likely to benefit these communities. And since then, there's been a lot more, many more versions of these guidelines, and they've become much more sophisticated. So the Golden Ridge hair sample has become part of good Indigenous science, but only belatedly. Haddon and Willislev, the two main characters in this story, the guy that collected the sample and the guy that sequenced it, eluded these ethical requirements. So the two main people just weren't aware of these ethics. For Haddon, it was simply a matter of time and mortality. When he was around, he was a good guy for his time, and he didn't have to follow any of these things. But for Willislev, national borders allowed him to spatially escape the ethical requirements that Australian scientists could not avoid. So he was told, I don't need ethics. But both scientists were considered good in their time. So even though we might think of Haddon as, typical race scientist, he authored a book called We Europeans in 1935, which bravely attempted to counter Nazi racial ideology. And Willislev, in turn, he experienced his Australian research, his experience being told he needed to get ethical or some kind of approval as an epiphany of ethical enlightenment. And he later delivered the most contested Indigenous skeleton in the world, probably, Kennewick Man, or the ancient one, back to the Native American tribes. So I'm just wanting to raise it. I'm not trying to say there's no clear good and bad actors here. Although there are competing stories about the exact sequence of events around this paper, it's clear that Willislev responded to this review of feedback saying, you don't have any approval, you don't have any engagement with Indigenous people. He responded with some top-level scientific networking. He made contact with a forensic biologist colleague in Perth, who contacted an environmental geneticist, who contacted an archaeologist, who contacted a historian, who was then research manager for the Goldfields Land and Sea Council, the native title representative body with jurisdiction for Kalgoorlie and surrounds, the place where Haddon collected the sample. So, and all these people are here. So the forensic biologist, the environmental geneticist, and then the archaeologist, and then this guy, Craig Muller, who's the historian. He became friends with SK and began looking at this encounter in 1923 and talking to the board of the Land and Sea Council, the Aboriginal Representative Council, about the possibility of SK visiting them. Craig Muller, this historian, concluded from his research that because the stop at Golden Ridge Station was no more than 40 minutes, and because Haddon was old and not physically intimidating, the sample of hair was freely donated, and therefore the Indigenous group could approve his use. So then SK made a quick trip to Kalgoorlie. That's him there with the Land and Sea Council. And they agreed to endorse the paper, and it was published in Science. So it was a happy story, I suppose, in so many ways. Everyone seems pretty happy here. But this just small little blip in publishing the paper tells us a lot. So this story illustrates how the ruins of one scientific era can become the spoils of the next. 21st century science brought new life to 20th century biological collections. Old hair collections posed a solution to the politico and ethical problems of accessing bones and blood. And this return to hair contains other returns within it. And this is an image from the magazine article accompanying the scientific paper. despite the political and semantic shifts from race to population in the mid-20th century that people like Jenny Reardon have written about, the first Aboriginal genome to be sequenced in 2011 was received as a racial story. And in the book, I go a lot into this particular image that was printed as Aboriginal Australian, like the one pictured above, and how the idea of a racial story was, again, returned to, despite this very new science that was used and despite the belated but still attempt to engage Indigenous community representatives. So this particulars of the encounter at a railway station in 1923 are relevant only as a media-friendly backstory, highlighting the cultural sensitivity of the scientist and the racial purity of the subject. Somehow, once more, a single characteristic hair defines a race. So I will leave it there, and thank you so much.
Thank you, Emma.
Thank you, Emma.
So we definitely appreciate your decision to focus on one of the main stories in the book, but I have to say that there are so many other things that are part of the chapters, and especially I want to start addressing the very interesting and subtle way in which you treat the issues of post-coloniality versus decoloniality in the context of everything that you say, all of this tracing and tracking of the history of indigenous biological knowledge production. So,
Paco, can I interrupt you 1 sec?
Sure.
There's a mirror behind you and it's interfering with how we view you because the camera keeps switching between the focus on you or the mirror. Can you turn your...
A mirror, you say?
You don't have a mirror?
No, I don't have a mirror.
Jeez, does everyone see him fine? He keeps...
No, actually it looks very regular to me, but I don't know if you're seeing something different.
I can probably try to fix that.
Never mind.
Thank you for the comment. But this is probably a good moment for me to stop and to announce in following what we promised. So next, Emma, we will listen to Vivette's comment on probably the presentation, but mostly, I suppose, the main topics and themes in the book. And as I said, in connection to some interesting interdisciplinary work that is taking place in Mexico City and in other cities in Mexico, and also in connection to scholars who are invested in these topics. Some of them are specialists in biological anthropology and other disciplinary orientations, and this is probably something that will be important in Vivette's intervention. And after that, we will start collecting some questions. I do already have some questions and comments, but we will try to prioritize the questions from the group that is joining us at the Zoom call, and also people who might be joining us in YouTube. So please, Vivette.
Thank you, Paco. And sorry about that interruption. I don't know why your camera keeps jumping at me. So thank you, Emma. I've prepared a very brief general comment and a set of questions that I think might help us to jumpstart the conversation. So also to attending to the invitation that Paco made the Red Balance or Balance Network to briefly state what we do. I'm going to share with you. No, I can't. I will share with you the link to our website in the chat. Let me do that before I continue. And briefly speak about this collaboration and how your book really resonates with some of the work that we have been doing. And I know you know this because we have also, you have collaborated with some of us on different occasions. So the Balance Network, the website is in the chat, and it's trilingual in English, Espanol, and Portuguese. We are working to think of ways of establishing collaborative research on human remains, and particularly on ancient human remains, with some of the tools that you've been pointing towards, the more contemporary genomics tools, in a way that reckons with the ghosts of racial science in Latin America, which we have many, but also with the geopolitical and disciplinary asymmetries and hierarchies that have been embedded in the development of these approximations. No? So in a way, we want to figure out ways of collaborating that challenge the usual received flows of knowledge production that are assumed to go from north to south. But as your book clearly shows, the materials that enter into circulation in a global economy of knowledge usually do not proceed from the north. They many times proceed from the south. So we want to trouble these ways of assuming how knowledge is produced and flows across these north-south polarities, and also think about how ethical guidelines for establishing work with ancient human remains are produced. Now, who produces them? Where are they produced? How do these norms interact with more local ways of handling, caring for samples, learning about materials that perhaps cannot be labeled as collections in the same way as collections in the British Museum are labeled, right? And also these materials that come together as a source of anthropological and historical knowledge that is contingent on the disciplinary unfoldings of these ways of interacting with them within national contexts that are very particular. You know? So this is some of the work that we're trying to develop in this collaborative network. We are a group of scholars from diverse backgrounds. Most are biological anthropologists. There are social anthropologists. There's an STS scholar, that's me. But I think you can find out more about our network there. And some of I'll try not to get into what the book and your work, Emma, has done for thinking about how to pursue these sorts of collaborations. So we've learned that from Emma Kowal's work that ghosts of racial science have not completely abandoned us. They are present in contemporary sciences, even in genomic research carried out by indigenous people with a focus on justice and reparation. These ghostly presences reveal something unresolved, not overcome in the history of science, which post-colonial approaches are failing to see. What is Kowal's proposed strategy? She says, rather than trying to exercise the idea of biological differences and distance herself from the white scientists in her book, which she could do, given her positionality and the ways in which she is approaching this object of research. But she proposes that we learn to live with the ghosts of past racial science and harness the evocative power of these very uncomfortable, hard to forget presences to raise questions about biological difference in other ways. So using some of the language that you used in your presentation, one might ask, if we are set out to promote the practice of good science, we should always ask ourselves, good science by whom and good science for whom? No? And I'd like now to put forward a set of questions that emerged during the reading of your work and in thinking of more methodologically about how to tackle these issues. So what would a history of science that takes on the challenge and the discomfort of dealing with these ghosts look like? So in your book, we see that this would not be a history that is articulated in a linear idea of time. So as if racial science were something outdated, it's already behind us, or something that we can completely get rid of. No, your book suggests, on the contrary, that the ghostly presences of racial science highlight entanglements between the past, the present, and also the future of what genomic science should or could look like. No, the term you use is that of folded objects. So could you tell us a little bit more about how you think about the spatial temporal folds that connect the past, present, future? And so one reading is that this is a methodological invitation to avoid two things. One, homogenizing the history of the biological sciences. So too much emphasis on the historical continuity that ignores the specificities of contemporary science and its tools. And on the other hand, assuming a radical discontinuity between current genomics and scientific practices of the past. So no reckoning with the continuities that do in fact exist. And you proposed a third way, no, where biological data can offer, you say, haunting possibilities and resonances. Could you give us some examples of these possibilities and resonances that have emerged from this research that are meaningful to you? No? It is also clear in your book that the materialities of the objects, so this story about this very particular lock of hair, has an important weight to identify and analyze the ghostly presences of racial science. No? And this is something that is not, this materiality is not something that is found in other ontological approaches, perhaps more sociological ones that you are in conversation with in your book, like Avery Gordon. No? So it seems valuable as a contribution of this work, like this focus on the more material aspects. No? Could you share with us how you arrived at the argument that these objects operate as haunting mechanisms? And what role does materiality play in the way you think of haunting as a methodological tool? In the first chapter, you say that ghosts have the capacity to disrupt our ways of proceeding and make us confront questions about what has been forgotten and erased. In what ways can a historian detect and analyze these interruptions? Is there an analytical strategy that has helped you choose and develop the analysis of the ghostly objects and practices that you address in the chapters of your book? So these are some of the questions that emerge and that I think many of us are grappling with when we try to reckon with these continuities, but not fall into the, this overall collapse of this is the same thing all over again. There are some particularities that we need to untangle from that work. So thank you, Emma.
Wow. Thank you, Vivette. And, Vivette, you're such a fellow traveler in so many intellectual currents that, it makes me feel very close to you. And that you have, people may know that is very prominent in the history of biology as well as STS, and or the history and philosophy of biology. Philosophy of biology, isn't it? More than history. Sorry, but just areas that I think we have parallel sets of concerns, and that's expressed in the just incredibly questions that really hit home, I suppose, that really get at what is important to me. So I will try and do a little bit of justice to them. How do you do history of science with ghosts? I mean, that's kind of a question that kind of goes through a few of your questions. And how did I get there? And it did kind of happen organically. I'm not a scholar that sits down and says, okay, now I'm going to use this theorist and in this way. I kind of don't believe in that, because I think we're humans and we have these enormous, like, glucose demanding organs of the brain. Maybe that kind of makes me a kind of biologically essentialist or neurologically essentialist, but I just think we have to, and all of our feeling sense organs. I suppose, again, I'm quite materialist about the intellectual process. So I really have been on this series of encounters with some of them completely random. So I'm trying to remember. Well, I mean, the story of the hair sample and the sequencing, I was, following these genetic researchers through as kind of ethnographic subjects for many years. The projects started in 2007, my first postdoctoral funded project, thinking about genetic researchers working in Indigenous communities. And so the hair sample, everyone was worried about it. Everyone was hearing, there were rumors that Eske Willerslev, who was kind of like a god who is there in Copenhagen doing all this amazing stuff, and that he had an Australian sample, and partly people were like, and I'm sure that people I was studying in my ethnographic group of geneticists were among the reviewers, so they all know about what each others are doing because of peer review, because of our intellectual production system we have in science. So that, and then the more I learned about the story and that it came from the hair, and the story kind of told itself. But you're right, Paco mentioned that it's not this particular story I told. The ghostliness is not the ghostliness is in the hair sample itself. Yeah. So, and particularly because I showed that image from the front cover of Science, which had the hair sample with an electron microscope. So and there were so many images. There was a whole lot of images of photographs of the hair, and then these more and more magnifying images. So and I suppose that in science itself, it's and the fact that it's the material and the hair and the DNA that can be traced to 70,000 years ago. So I am kind of partly, I think it's part of being an anthropologist of science, an anthropologist of anything, that you kind of enter into the magic making of whatever your subjects are. And, for scientists, materials make magic. A lab is a place where, you make materials tell their secrets and interpret them. So I suppose I'm just illustrating my interest with materiality rather than explaining it per se, but maybe that'll have to do. But then just this question, and this second kind of question about living with ghosts. And as you say, and I kind of laid out that I suppose I'm very contrary. And when anyone thinks that they have an answer, I'm like, are you sure? Are you sure that's the answer? So equally, and I came from it from, being someone working in Indigenous health and in Indigenous solidarity activists who I felt like, oh, of course, the answer is that we just need to, institute self-determination of Indigenous people, and that they decide what healthcare they want or what research we need. And if we have Indigenous people deciding it themselves, then that will solve the problem. Because we are so different from people that came before who were racist and did not really consider what Indigenous people wanted. Now we're much better now. And so this is and actually in my first book, I talk about historical discontinuity as being like what the white anti-racist subjectivity depends on us being different to the people that came before. And of course, this was something that I was, critiquing. But then equally, within the social sciences, scholars who were just like, looking for the race, looking for what, where the racism is, and okay, here we've found it. There's this new scientific practice, but they are racist like the people before, even if they are, culturally diverse scientists themselves, and so this kind of like constant desire to kind of find the historical continuity and say this is just like it came before. That seemed really unsatisfying to me, because these things are different, and the ultimate illustration of this difference is that I have been, part of building, and there is very much a strong institutions that are Indigenous led. So in Australia, we have the first professor of Indigenous genomics in the world is in Australia, is an Indigenous doctor and scientist. And then for me, it makes me, I don't want to be like sour grapes, like the person who's always going, well, even that's not good enough. So it's not about whether it's good or good enough. It's that these questions of, how do we understand difference? How do we conceptualize difference in a way that is, empowering, not that doesn't reduce us at all, as people, as scholars, as people that want to see justice. These are all questions which I think, if we think that we have solved them, then we definitely haven't solved them. So that's the kind of living with ghosts, that wrestling with ghosts that I want to advocate for.
Thank you, Emma.
Would you like to respond to Emma's comments, Vivette, or should we continue?
I think we should continue to give other people a chance to participate.
Right. So we still have at least 40 minutes to go. So please, if any of the members of the Red Balance would like to now ask something to Emma, and also our other colleagues from Surge. I see now that Carlos Lopez Beltran has his hand up, so maybe we can listen to Carlos and then start to gather more other questions and comments from you. And also Jimena will take care of the YouTube contributions. So Carlos, please.
Hi, Emma. How are you?
Good to see you. Yeah, I saw you in Oxford.
Yeah, about a year ago. Yeah. I'm a bit embarrassed because after that talk, I got hold of your book and then I didn't read it all. So I still have it there waiting for me. But so probably the question I'm going to ask is already answered in there, and I apologize for that. So it has to do with, this guy, Erske is his name. He was here in Mexico. Probably Juan Manuel can tell us more about his visit. And it was a very interesting meeting that Juan Manuel, is he still around? Yeah, because I don't see.
Yes, I'm here.
Hola, Juan Manuel.
In the
organized where he brought to Mexico some of the big shots of international ancient DNA research, and basically the questions and Juan Manuel wanted us to discuss in that meeting had to do with who should be using the samples that he was in charge of ancient DNA, of ancient bones that are collected are accumulated in the National Anthropology Museum. But what was interesting is that there was a tension all the time in the meeting between doing things ethically and properly, and discussing how to do that. So, and the you mentioned it, the competitividad. How do you say that?
Competitiveness, no?
Competitiveness. That's the word of these guys. Reich and Erske and others that wanted to get hold as quickly as possible of the samples because they wanted to win a race or something like that. And so my question is about, is that any different when you introduce indigenous interests, indigenous researchers, as you mentioned, into the or when the research is done with their participation, you still have this same rat race, and you still have this same competitiveness and this economy of getting there first, and I'm considering whatever the solution is, or the answers to scientific questions are going to come out, just general objective knowledge that belongs to humanity and not really to local people. I don't know if my question is making sense, but it really is about what changes if you introduce these other actors really in the way science is, or are they really different questions, or how different are the questions? Anyway, that is.
Thank you, Carlos. So Lumila will be next, then Pepe, then me. Emma, please, if you'd like to address Carlos's comment and question.
Yes, well, I've just put in.
But I think Juan Manuel wanted, to say something about Carlos' intervention.
Probably, right? Probably Juan Manuel could clarify some of the issues. Yeah.
Okay. Just to put the things in a context, because Emma, you talk about the schedulers that work in 2010, and the thing is that I was the director of the biological anthropology department in 2015. or 2016 maybe. And then when he started to do this, big works with DNA, because he was, I'm sorry, making this sophisticated methodology. He started, and also David Reich, and also I must say Krause from the Max Planck Institute. They started at the same time that they was making advancement in the methodology. They started to ask for some biggest kind of samples to Mexico. So I was the director, like I said, so I was very, confused, because it was like, can we have, 2,000 samples, 3,000 samples? We were talking about thousands of samples. So we decided to, cut out the samples to them. And then we have a meeting about the, why they was doing that. And Carlos was in the meeting, Paco was in the meeting. So to put it in context, it was a very, rough or very uncomfortable meeting, because they actually didn't understand why we have a problem about the size of the samples, and they actually did not understand that we have a stock of human remains, and a big difference between Australia and Mexico is that some of that indigenous persons doesn't exist as a group already. I mean, there's not like Aztec people or Tlaxcalteca people, because it was a different history. So we actually are I don't like the word, but the stockholders, and and we have the responsibility of all that samples and all that. Sorry for say samples, all that human remains. And Esquiveros was very aggressive about that. He didn't understand what was the problem, because he says like, this is for the advancement of knowledge. And the advancement of knowledge, it's always, a first world favorite, kind of sentence. And here we are more, occupied in rescue the human remains and the patrimony, that to make the advancement of knowledge. So it was a very different kind of way of think. So actually, David Reich and Esquiveros left mostly. Those guys didn't understand our position, our ethical positions, and right now, and this is my, and right now, they still don't understand, because NACRA, it's, I mean, they have guidelines, but we have guidelines, and everything is around, the guidelines of those guys. No. They are not attended our guidelines, and our guidelines are made for our, ethical, made of handle the human remains. I don't know if you understand me, or I'm just babbling because you can know my lack of English.
No, yeah, absolutely, and it's really interesting, and also, I don't know, disappointing to hear that was your experience in 2015, 2016. Definitely to the outside. So I actually, in the chapter, I quote from a New York Times profile of S.K. Willislev that was in 2016. So I suppose just at the time he was talking to you in Mexico City, he says in this profile, or the profile tells us, his experiences in Australia, SK's experiences in Australia, have changed the way Dr. Willislev and his colleagues investigate DNA from Indigenous people. I've evolved, he said. So, and which is, a beautiful play on words, of course, because he studies evolution and he himself has evolved. He accounts from his account from this experience in Australia. And he's telling, the New York Times that he has evolved. But if at the same time, if he's in Mexico City, not very evolved with his views, that's disappointing. And I suppose, it does lead someone to be pretty cynical about his claims to have evolved.
And I need to add that meeting was organized by the National Geographic, because, of course, when I got the samples, they say we have a meeting with these guys from Mexico, and today we don't have the tape record of that meeting. So they do the meeting. We do the meeting, but they organize all the fight, because it was a kind of fight. And today we don't have the record of that reunion, and they do an inform that is really not similar to our experience.
Yeah.
Thank you, Juan Manuel, and thank you again, Emma. Definitely, when I have my turn, I'll say something else about that, because I attended that meeting, so there are a few other details that might be of interest for you, and also maybe, if I may say so now, to think about a potential collaboration in terms of comparing these experiences from these scientists in the Australian context with the Mexican one. But it's Lumila's turn now. Please, Lumila.
Thanks, Paco. Well, thanks a lot, Emma, for your talk. It was really interesting to listen to you. I mean, I'm familiar with your work, with your papers, but then it's nice always to listen to someone in first person. So thank you. I have a brief comment and two small questions. The brief comment is that actually, as part of the Balance Network, we are preparing a book chapter. So we contacted you. It was me and Constanza de la Fuente. We contacted you for the special issue we are organizing. So yeah, you will soon hear more and learn more about what we're doing through the chapter we're preparing. So that's a brief comment. And then going to the questions. My first question is brief. So you very accurately use the word biological differences to describe what researchers or what the people were investigating in the end of the 19th century, beginnings of the 20th century. Because I think, yes, it's true, they were really focused on the biological differences. But then we know that there was a switch during the 20th century in the history of the biological sciences, but also the biological anthropology, towards, instead of focusing on the differences, we are more focused until today on biological variation. So the first question is, how do you see this change affected positively, or I mean, is just a conceptual issue, or do you think, no, the other way around, is it just a semantic issue, or do you think there is a concept there that changed the way of studying the human variation in a positive way? So how do you see this connected also with colonialism? And the second question is related to Latin America. So you mentioned that there is this person that became the first professor in Indigenous Genomics in Australia. We also know that there are a lot of people in the US, in Canada, so still countries from the global north, in which we can see that people from indigenous communities get access to academic environments and to work in academic environments and to do research. But reality in Latin America is very different. So indigenous people are intersected by very difficult socioeconomic issues, very poor socioeconomic conditions. And I would say, I don't want to generalize, I'm not going to say all, but most of them cannot really get access to high education, to the university. So how can we think about positive changes when we compare that with realities that are completely different? And what can we do? What do you think could be some tools or some ideas, also in terms of practice, in Latin America to take things forward, one step forward?
Great. Thank you. Yeah. So, well, on the question of what to do in Latin America, I think Pepe's, who I know is involved in Mexico as well, might actually address that when he speaks, because, I think that I mean, I don't know the particular statistics in terms of Indigenous people in Mexico, and I know, of course, in Latin America, the kind of the definitions of these terms do not always hold, but it is also the term indigeneity and the concept has forever expanded since it kind of came into being, really, again, in the middle of the 20th century, and initially through the International Labour Organization. So that's also really interesting to see, where does this term Indigenous even come from? I mean, materially, biologically, Australia is like the best case for the term Indigenous, in that there were people there for tens of thousands of years, and then Europeans came extremely recently. But in most places in the world, it's not. It's pretty complex in terms of who was there and who came. I mean, in Latin America, you've had such a much longer history of European colonization. So it's complicated, but given that indigeneity is forever expanding, at least the last 70 years, it's continued to expand to places like Indonesia and India, definitely Taiwan. So it's kind of expanding. And I think in Mexico, absolutely, I think it is probably increasingly salient as a concept. And so then things like the summer internship for Indigenous genomics, which absolutely has come from the global north. It came from the US, then it went to Canada, it's come to Australia. There's also a Sing Micronesia that is, I think, has had their first one already. So that's kind of one question. So, and I think probably Pepe, I don't think Jocelyn has made it, but they would be good people to talk about that. The question of difference versus variation, I mean, this is probably where we need Vivette, the philosopher of biology. And there are some really amazing Australian philosophers of biology. Paul Griffiths comes to mind, who can probably better answer this question. I can really only just comment on where I think my own use of these terms is going. But so, difference versus variation. I mean, one way to say is that, racial difference was what the 19th century, early 20th century scientists were. So we kind of can put difference with race, and that there were, because they were looking for very sharp actual differences to put people in, clumps in specific groups. And then we shifted really from the mid-20th century, and, Jenny Reardon in her book, Race to the Finish, and many other people talk about this shift from race to population, and a shift from difference to variation. So now we're looking at variation within a population. You've got populations that overlap, where there might be, quite overlapping, but there's still some parts that are not overlapping. And this is this is what makes us different. So this is the historical discontinuity for scientists. Like the bad scientists were looking at race, we are looking at population. They were looking at difference, we are looking at variation. So you're probably not surprised that I don't really buy that as a distinction. I mean, I think what's interesting is the effort to make these distinctions. Like anthropologically, I'm thinking, okay, how are you trying to make ourselves different? And not to say that there definitely are differences between saying, a single hair, that quote from Bruno Bay, that a single hair can define a whole group. That's like kind of pure race theory. but even when we are looking at variation, and we've seen this with like personalized medicine, precision medicine, we are looking for the differences, even though of course we know there's variation. We're looking for the differences. And eventually we're looking for the precision, the actual differences of this particular person. And we're not interested in their race. They're interested in whether they have, this CP450 variation or not. And then maybe that's fine. We've kind of moved beyond race, but actually there's a paper, I'll put a link to it, that I've done with a evolutionary geneticist friend, Bastian Llamas, about long read sequencing, which is, again, this, it just shows that even when we are getting towards this horizon of we no longer need to talk about human differences at all. We don't need to talk about group differences at all. The idea of the group just always comes back. So I'm not sure if I answered that very well, because it's a huge question.
Yeah, thank you. It was just the idea was just to share thoughts, and yeah, that was great. Thanks.
Francisco, we cannot hear you, but I think you were giving the word to Pepe.
Yes, that's right, Jimena. Thank you.
Thank you very much, Jimena, Paco, and Emma. It's a pleasure having the opportunity to listen to you again. Unfortunately, I'm not directly involved in Sing Mexico, so I cannot answer the questions either. But maybe Jocelyn can see the transmission on YouTube of this conversation, and then get in touch with everybody involved just to make a bigger collaboration network. But for now, I would like to ask you, Emma, in your opinion, to what extent do you believe that these indigenous governance structures, such as those implemented through the National Center for Indigenous Genomics, can meaningfully mitigate the political risk associated with genomic research? Even when the research is community-led, how can indigenous genomic initiatives safeguard their findings from being co-opted by state actors or political agendas, as seen in cases like Mark Latham over there in Australia, or in the United States by moments in political history of our neighbors, such as the one that was I'm lost at words, but the one where Elizabeth Warren was involved, and that she has received a lot of criticisms for that. Do you think it is ever fully possible to protect genomic knowledge from being misrepresented, reinterpreted, reappropriated, or weaponized against the very communities it is meant to serve? And even though your last intervention started to answer a little bit this question, I was taking notes. I would like to ask you, in your opinion, what responsibilities do we hold as scientists, as educators, as trainers of the next generation of scientists to foster an ethic of care and accountability in the production and use of that genomic knowledge? Thank you, Emma.
Thank you, Pepe. It's really good to see you again. So this is a big problem, which I think is I can't see a solution to this one. So the issue of the misrecognition or the deliberate use of information about Indigenous biological difference, Indigenous genomics, to the detriment of Indigenous people. So you mentioned this, I talk about in the first chapter of the book, Mark Latham, who was an Australian politician who in the like lead up to an election I think it was like 2019 maybe, was proposing, he's from a right-wing party, although he used to be left-wing, has become right-wing, and he said we should limit benefits to Indigenous people to people that are at least 25% genetically Indigenous. So this has obviously caused a huge media storm, and my colleague, who was he's the most senior Indigenous scientist in Australia, and was involved on the board of the National Centre for Indigenous Genomics, was in the media saying, we don't have the science yet. We're not able to tell someone's percentage of indigeneity. And other Indigenous people are saying, no, this is a completely irrelevant way of that Indigenous identity is not biological, and it's also very racist and harks back. It echoes previous eras where the government was decided who was and who was not Indigenous. So all of this kind of discussion was swirling around, and that it's hard to. I don't know what's preventing that from happening again and again. The question of false identifications, which is, very, very much a ongoing issue in the US and Canada, in that it seems with quite a bit of regularity there's like a identity scandal, like someone, particularly in academia or in other kind of prominent positions, who's been saying they're Indigenous, is then people say, oh no, they're not actually Indigenous, they don't have evidence for that. And sometimes it turns out that they don't, or sometimes they thought they were Indigenous but they weren't, or that, I'm sure there's many, false claims of being false. But this is something that I'm also interested in, because it flows, and the reason why I say I don't think it's going away is because direct-to-consumer ancestry testing is now available for Indigenous ancestry. So, and this only happened in 2020, that Ancestry DNA changed its algorithm to offer a part of their algorithm is Aboriginal and Torres Strait Islander ancestry estimate. And I've been following this. I've actually applied for funding to look at this more, because there are 20,000 customers in Australia. There's over 1 million customers in Australia of this one company, but over 20,000 have got some Indigenous ancestry on their results. And we don't know, from following like social media groups, I can see that many of them are going then to these groups and saying, I've got this result, what does this mean? And tracing their ancestry, their family tree, their genealogy. So it's just another incredible contradiction in this. So at the same time, when I feel like I spent a lot of my younger years saying, yeah, things are not biological, indigeneity isn't biological, we are now having both, Indigenous scientists saying, we need our own Indigenous genome, and, publishing work on this. And then also Indigenous people, or people who do or don't identify as Indigenous, getting genetic tests and really, really embracing that and trying to find, what is their connection to their, great grandmother who was Indigenous. So I can't, it seems to more and more biologization, and it's very dangerous, very easy to be weaponized against Indigenous people. So I don't know what to do.
Thanks, Emma. So I think it's my turn now. So there are 3 things that I want to mention, and I really need to be as brief as possible. the first two things that I'll mention will be only for us to maybe think about them and continue the dialogue and the discussion later. And the third one is one that I'd really like to start discussing with you, Emma. So the first one has to do with, I'll pose it as a brief question. So is molecular genetics-based ontology compatible with indigenous ontologies in plural? I think this is something that appears in several places in the book, given the interaction at the social level between the indigenous communities and the scientists working on genomics in general, right? Because in principle, and also being most of us partly anthropologists ourselves, we know that the way in which outside of the West, the relation between, for instance, nature and culture is constructed doesn't necessarily imply that DNA holds the key to life, right? So I think that one of the many very important threads of discussion that your book establishes is this one. No? For the particular case of Australia, how do we see answers to this particular question? So the second issue has to do with actually the history and the philosophy of the life sciences during the 20th century. So if we think, for instance, in relatively recent accounts of what happened during the 20s, 30s, 40s, not only with the modern synthesis, but also with the demise of organicism, for instance. No, I'm thinking, for instance, of Eric Peterson's book, The Life Organic, no, where he talks about, Warrington and Needham and all the guys who were living into an organismal biology, not necessarily a biology that was to be based or dominated actually by a molecular genetics frame of mind. No, so I think this is a topic that could be actually discussed as a derivation of many of the implicit ways in which you tell the history of biology in the 20th century. No, what would have happened if molecular biology as such, and then later on, contemporary population genomics of humans and of any species would not be the dominating way of looking at things, no? And how is that related to our current interdisciplinary concern in issues related to DNA and ancient DNA in particular, no? So if biology were more Warringtonian than Watson-Cricksonian, if I may say so, what would have happened? No? Because also I think this question is important for one of the main issues that you wanted to discuss in the book, which is how do we understand human difference? No? So because if we think about human difference in terms of epigenetics and niche construction, for instance, the issues that are on the table are different than if we continue to assume that something in the molecular genetics of humans and any other species is really the most important thing to pay attention to. So these are only two things for us to continue discussing later. And the third one has to do actually, I will start by my own recollection of what happened at the meeting that Juan and Carlos were talking about. This was held at the Marriott Hotel in Mexico City, so actually very close to downtown Mexico City. As Juan recalled, the National Geographic Society was the sponsor of the meeting. Bernardo Yáñez, who is now in the call, was also present. So you mentioned the Rasmussen et al. Paper twice or three times during the talk. And we could see the list, we could recall the list of authors. So there were at least two Mexican authors there, right? María Ávila Arcos and Víctor Moreno Nayar. María and Víctor, young, very bright and accomplished Mexican genomicists, were at the meeting, and I to this day cannot avoid, especially in the case of Víctor Moreno Nayar, associate him to the way in which pharmaceutical companies, for instance Pfizer, were actually very enthusiastically putting money for the financing of projects related to genomics and ancient DNA research in particular. So the issue of the connection between all of this ancient DNA research and companies that for decades have been pushing forward biology in a certain direction is, I think, an issue that needs to continue to be discussed from this starting point that your book and other books like yours have now deployed and have actually given us for as material for discussion. So I would like to listen your comments about the connection that you make in the Australian case between, molecular biology, if I may say so, as a capitalist business model, and the science that is produced and the information that many different actors, stakeholders, if you want, are getting to be used in their own terms, for their own purposes, for their own interests. Thank you very much, Emma. This has been a wonderful session, and I'm sure this will give us plenty of material to continue. And really, I hope that at some point you can come to Mexico City to continue this conversation.
Oh, thank you so much, Paco. I'm just so many people in your group there at UNAM that I love to be talking to. And I love this set of questions. And I think maybe there's some overlap I see in the questions themselves, which I think comes back to this ontological difference question. So do molecular genetics ontologies and indigenous ontologies, how different are they? Is their differences consequential, and how do these differences, express in terms of, one over the other? And actually, when you were talking, I think about, we've got, Indigenous people are people, and Western people are people, and I do think that the human factor is overrides everything else in the end, which is, not to say that there aren't differences. And, I'm an anthropologist, so my whole, disciplinary identity is depends on there being, important differences to understand. But, in the end, humans. So it made me think of like native title, which is a form of land tenure in Australia that we have developed really in the 1990s to try and recognise particular Indigenous rights in land. And people like Elizabeth Povinelli have written really insightfully her book, which is quite a while now, the politics of recognition, the cunning of recognition. Sorry, her book there, really classic analysis of how this recognition of Indigenous ontologies of land ownership and Indigenous histories are recognised within the Western legal system, and how Indigenous, similar to Brazil, not really so much Mexico, but in Brazil, the whole, like in Australia, anthropology is very substantially, its job is to record genealogies, record, families and who owns what land, to then be part of a, Western legal process. And it's some kind of melding of, recognition literature, which I've always found really, really useful for this kind of, for the multicultural, settler colonial, post-colonial nation. So there's a recognition, and there's also, there's a mutual recognition, and there is Indigenous people, whether they're, native title leaders, leaders of their groups, or they're trying to, become lawyers, enter the Western Academy. They are, they're engaging with the rules of the game, the tools that are available to them. So I suppose I'm not. I suppose I'm not as like hopeful. In some ways, this goes back to my, I don't know, you might call it, very persistent cynicism about any kind of easy answers. And I suppose I've seen, I've had decades of these kind of call to indigeneity of like, well, we just need to look to Indigenous ways of doing, and that will solve it, that there is some kind of, salvation. There's some kind of way out. And I suppose I don't have very much patience for that anymore. Maybe that's just becoming, old, grumpy middle-aged woman, which, I'm willing to own. I'm willing to own that. But just, because I just, you see cycles and cycles and cycles of like, well, here is the answer, here is the answer. And I talk about this a lot in my first book, either we're just really bad at trying to do the right thing. And, with the world today, that is the most obvious answer, that we do have this capitalism gone completely crazy, and even, democracy is incredibly under threat. So so in some ways there is so much that needs to be done to make the world even, remotely livable place. But I suppose in work, I've focused on where people are trying to do, the right thing, trying to do good science, and where there are resources to support that, what then? So I suppose I'm interested in not where there are really obvious things that are going wrong, so with SK and David Reich coming and being very insensitive, perhaps, is how I'm hearing that, how they were behaving, and, like, oh, well, we need more samples because we have to do great science and that's for the benefit of the whole world. And, how dare you try and stop us? That is clearly bad, and they need to be different. But I suppose what happens when, and I think what you were saying, are we reimagining an organismal biology? And maybe that, and I love those counterfactuals of like, how could it have been different? And I mean, it's quite interesting that I think the kind of counter argument would be, well, genetics is naturally moving towards epigenetics. So, and I've just been on a panel reviewing grants in genomics that are trying to improve health, and so many of them are about not looking at the genes, but about epigenetics, about the expression of proteins, because the genetics by itself tells us, not enough, so little. So in some ways, that kind of becomes a kind of science as usual, that the science will continuously just reach towards this organismal biology, and we see that, I would argue, also within ancient DNA, that we've gone from like, okay, we've got a few little DNA samples to really moving towards, ecology, and potentially, AI, again, it's a kind of technological salvation, but also potential horrible apocalyptic outcome. But on the happy side, AI might just be able to figure out the whole history of humans, and, all of the different, hominins that we don't even know about yet will just kind of all emerge from the data in a holistic way. That's very overly optimistic picture, but all of these are possible. I don't know, but I'm excited to be watching it all happen with you, Paco, and we can keep on analyzing it.
Sure. I hope we can. Thank you very much, Emma. I'll give now the chance to Jimena to lead us into the conclusion and rounding up of the session.
Thank you
If
very
there are
much,
no other
Paco.
questions.
Thank you, Paco. Thank you, Emma. As you can see, we can go on for a little bit, and we are very grateful for your talk and answering all the questions. There's a lot of very interesting topics on the floor, on the table, or whatever. There's just one question on the YouTube channel, but I think we already talk enough about this point, but I'll still read it because it's from one of the social services that support Surge. Thank you very much, Mauri, for listening to us. And maybe this can lead you to a final comment and closing the session, right? Entonces, Amaury is asking, what does scientific work need to do to overcome its colonial profile beyond the ethical shift that prioritizes the approach between the subjects and the objects, which are many times subjects also of research? And as I said, I think we already discussed enough about the possibilities and limitations of genomic science work. But, well, maybe you can just give us a closing comment. And again, thank you very much for everything.
Oh, thank you. Thank you, Jimena. Sorry, was I? Yeah, just,
no, please go on.
Yeah, I mean, this is just, continues to be an area where there is so much bad practice that's going on, and so much using samples without permission and without engaging communities, and this it shouldn't happen. And I think there was this, I think maybe it was Pepe before, I don't think I fully answered it, but the what happens in Australia is that things go very, very slowly. So things will take a lot longer to get published. And yeah, David Reich and S.K. Willisov, like, they may be collaborators on these papers. But they're not going to be leading them, and the samples are not going to be. They're going to be under the control of the Indigenous groups, who are often the scientists as well themselves. So so it is not an area where there's going to be a huge amount of scientific progress very quickly. It's going to take a long time, and it does take a long time. And so that, SK, the Willisovs and David Rice and the Johannes Kraus, they might be used to, getting a sample, and then, within 12 months, they've got a manuscript, or even faster than that, I've seen. But it's going to be like 5 years in Australia, because just every step we'll need to go back and talk to the community if they want to, actually we want to do this other kind of analysis now, or then just go backwards and forwards, and then how are we going to publish this? What are we going to say about it? And always be community members as authors, which we didn't see on the Rasmussen paper back in 2011, but now you'll always see that. So yeah, it goes very slowly, and that's okay.
Thank you, Emma. Really
Paco, do you want to say something to close, or Vivette?
Yeah, I think I can say that first. We also, besides thanking Emma, we thank Vivette and the group of Red Balance that gathered today, as well as some of our colleagues from Surge. I think it's quite evident from what we just started to discuss that there are still many issues to be approached and addressed in the case of Mexico, and the connection, the interaction between notions of indigeneity and the social actors that in Mexico claim indigenous ancestry, right? So, and considering that some of them are willing, and we have some data regarding that. We don't have time to discuss it now, but definitely it is something that we could talk about later. Some of these actors are willing to use some of the information from genomics and bioinformatics to continue reconfiguring their identity. So this is something that I actually think is connects the content of Emma's book with our situations here. No? And maybe this is the case for some other Latin American countries. And precisely I sense that some of the issues that this research group led by Vivek is dealing with have to do with this, among other topics, right? So all of the other things that we mentioned regarding the history and philosophy of the life sciences during the 20th century are quite interesting to me in particular, but maybe we will have to leave them for separate occasions in the future. And really, we are hopeful that Surge, and also thank you for all the people who make these transmissions and connections possible on the technical side. I continue to hope that Surge will continue to support these interactions, these international academic interactions, and so that we can see the light in the many difficult and complicated issues that Emma very kindly addressed for us this today. I didn't know if I should say morning or afternoon, but probably both, given the time difference between Mexico and Australia. Thank you very much, Emma.
Thank you. Thank you all. I look forward to more conversations.
See you next time.
See you next time.
Thank you.
Thank you, everyone.
Ciao.
Muchas
Thank you, Emma.
gracias.
Forum.
Gracias, Jimena, como siempre.
Con mucho
Gracias.
gusto.
Saludos a
Gracias a
todos.
ustedes, Vivet, Paco, Carlos, Karina.
Gracias,
Emilia.
Jimena.
Gracias, Monse.
Mil
Chao, Emma,
gracias.
gracias.